3 white flowers on that one. You probably got several hundred purple flowers on this one. So you have a lot more potential here in order to do this. Now these are both what we would call PI lines. So these aren't the kind of soybeans you would grow in a farmer's field every day. But what is cool is that they have a lot of different genes and traits that we probably don't see in our soybeans all the time. So in the United States, the USDA keeps a collection of all of the known soybeans that we have access to here in the US. And there's a little under 20,000 of those different lines. Whereas all of our elite soybean lines, like the stuff that you would grow out in your farm or your field, like Pioneer or any Corteva company, your Bayer, there's really not a whole lot of diversity in the soybean roots that you see in the field. You might see some diversity in leaf shape, leaf color, a little difference in yield, but the roots all look pretty similar. So that kind of tells me as a scientist, hey, that, you've probably seen this bottleneck effect where we have a few roots that have really kind of made it through this selection pipeline over the years, and we don't know if we lost a lot of the useful genes that could be really helpful for roots. So something that I can do and explore is I can start crossing soybeans that have really strong and healthy roots with maybe kind of some weak and measly roots and investigate which traits actually start appearing. If I see that whole spectrum of offspring that they make, from everything from I'm a really good root to, I kind of suck a little bit, you can start to see all of the differences in between and investigate where those traits are in the genome. So Chitiri is going to talk a little bit about using CRISPR-Cas. And that's something that I can use too, where I have this technology in genomics where I can actually go into a genome, and once I think that maybe I have a gene that gives me a really deep taproot, I can use this CRISPR-Cas technology to knock that gene out or to turn it off, more or less. So I break the gene in the DNA. And that allows me to say, hey, what happens in the soybean root then? Did that taproot stop growing as deep as it used to? And I say, hey, it did. All right, this must be a gene that helps grow for deep taproots. So now Dr. Singh can start looking at that gene and say, hey, this was a gene that we didn't have in our normal germplasm. I want to bring that in and hope that our elite materials now starts having even deeper taproots, giving me potential for stronger drought resistance. So there's a lot of opportunity when we start investigating all of these different diverse soybean genotypes. And it's really hard when you just start doing it on your own, just looking at it one by one. Yeah, I can kind of see some differences here. It's pretty easy to see it. But how do I quantify that? How do I actually take measurements? Yeah, what do you got?
Can you talk a little bit about the process of breeding soybeans?
Yeah, for sure. All right. So when we're breeding soybeans, and Dr. Singh can probably touch on this too, but some of the things that we're going to start looking at is when I am crossing one parent with another, what are my desired traits? What am I trying to look for from one parent versus the other? So take my dad, for example. All right? He's a really athletic guy. Okay? He was the kind of guy that he would just sink three-pointers all day long in high school. Okay? My mom, she was definitely the person sitting in the books and reading through them all day. And myself, all right? I have the potential to pick up any of those traits, but I'm kind of in science for a reason. I really suck at shooting those three-pointers, and my dad's still really good at it. But I really enjoy digging through the books. So when I'm looking as a soybean breeder, I want to say which traits do I want to have the most option for? And I'm going to pick those parents. When we start doing that, maybe I have a line here that has, let's say, really good secondary roots if I'm breeding below ground. And then I have, I can maybe use that plant over there. It's got really good taproot. And I say, all right, I'm going to try and cross these two lines. And when I would do that is I would actually look at these itty-bitty tiny flowers here. And actually there's one right there that would be ready where we could open up that flower. You can see that, not that purple one, but the one next to it. We would actually open that flower up in the crossing block, and we would take pollen from the other plant, and we would very carefully with tweezers put that pollen inside of that flower. Because soybeans are naturally selfing plants. So that means that by the time you see the flower, they've actually already they've already pollinated themselves, and you really can't do anything because the pollen has made it into the plant. It's ready to make those actual soybean germ inside of there. So we'll cross that. And then once I get that, we'll make a lot of those attempts at those crosses, so crossing the two different parents. And then we start looking at all the offspring and say, all right, which one of you offspring has the traits that we're looking for? And we keep you. We select you and we advance and we move you forward. And every year we keep testing that. So Dr. Singh, he starts looking at a lot of yield in combination with protein. So he's trying to look at okay, what parents can I combine that have really high protein traits? And the ones that have high protein and good yield, we keep moving those forward. And the ones that don't, oh, sorry, they get left behind several generations. So it typically can take anywhere from 6 to 8 years from when you start making that cross to when you do all of the evaluations, and then it could actually start being released to farmers for them to grow in the field. So does that help answer your question?
It's behind, it'd be like slower than corn because corn you're getting the whole ear worth of seed versus the soybean you're getting 2 to 3 seeds.
So the statement that typically soybeans are a little bit slower, yes, because corn has this awesome technology called double haploids. They can do some good genetic stuff that way. But typically when we're looking, we use a modified system, but you're advancing 1 or 2 seeds at a time and planting those out, and then you're going to see what those offspring are, and you maintain each one. So we do a little differently where we'll bulk all of those together. So you're right, you can have a quantity difference, and that can help in your numbers. So if you have a lot more corn seeds, maybe you have more to pick from. But for ours, we won't start making selections until probably that 3rd or 4th year, because right away, again, you don't have enough seed, and they're still segregating. So the genes are still moving between the chromosomes until you get a pure line. So yeah, you do get more seed, but it's not necessarily that gives you the time advantage. Does that help? Awesome. Yeah.
We do start selecting it F2.
Yeah, so yeah, F2.
Just for clarification, we start selecting as soon as possible.
Yeah. So the ones that are surviving and making it through. Yeah. What traits would you start selecting on your F2 versus later generations? What are your first traits that you're looking at? One thing that is really useful, and Chatiri going to talk some more about that as well, is you can use markers in a genomic system. And you can say, all right, I know that I want these genes to be in the plant, and when they're at the F2, I can actually use that marker and analyze, is that gene there? And if it is, I keep it. If that gene isn't there, I chuck it. So I can select there as well. And you can do that early on. You can do that pretty early. So, yeah. Any more? Awesome. So that diversity is really important as we begin to look at roots and we start breeding below ground. Now, another cool thing that Zachy has done is we're able to investigate all of the structure of those roots. So I can take a picture of these roots. Oh. And we can run it through what we call the advanced root imaging analysis. And so I got a picture of a soybean there, and the computer then will be able to find the taproot for me, all the secondary roots, and skeletonize it so that I can start pulling traits out of here. And I can start comparing that with a computer instead of me trying to do it by hand, tape measure, and actually making all those decisions. So again, I'm not an engineer, but taking certain engineering classes and working with engineers enables me to understand what they're doing and the thought processes that they're using. And then I can have really good conversations with Zachy and say, hey, what if we use this kind of approach to try and acquire these traits? Which really allows us to make better decisions when we're trying to investigate a lot of different soybeans and all of that diversity. So you can see there. So has anyone in any of your classes investigated or talked about machine learning or engineering or even talking about code at all in any of your classes? Anybody? No?
So Luis is their TA.
Okay.
And Luis will be pulling in a lot of information. And he's a great person to ask on some of that sort of thing with as well.
Yeah.
So all the solar data and the sort of pieces that they're going to look at.
Awesome. So hopefully you'll have a little bit more conversation about that. But again, when I was an undergrad, I didn't have any experience in that. But it was something that I was able to investigate and say, hey, this is really useful as a plant breeder and as a plant scientist to help me get more data, more consistent data, and be able to use that to make more informed plant breeding decisions. So you've had some good questions about roots, about breeding. What other kind of questions do you have so far? Related roots, breeding.
Are you also using for nodules for like a soybean cyst nematode for disease resistance or disease assessment?
So that's a really good question. So she asked if I'm using it for soybean cyst nematode. In the images that I have, I myself can see soybean cysts on the roots, but we intentionally trained Snap not to identify the cysts. That way we only get the nodules. However, it would be pretty easy to train it and then so you can use transfer learning then, and then train it to find just the cysts on the roots. You know, fortunately, there are pretty good systems to get cysts off and to count it. I know Sarah's done a lot of that in her past. But you know, in combination, then we can actually start investigating how maybe do cysts affect nodulation, maybe certain genomes that nodulate a lot try to compensate if there's cysts there. So you know, you can ask a lot more questions with that technology. So thank you. What other questions are there? All right, well then with that, I'll pass it over to Chitiri here, and he can tell you some more about some of the molecular and genetic things.